Once upon a time, in a land far away, a baby zebra was born. However, this baby was not your average colt. Instead of having black and white stripes, he had rainbow stripes. The little rainbow zebra's parents were confused. Why did he look different from the rest of the herd? The parent zebras took him to the local zoologists, enlisting their help. The zoologists told them not to worry. The little rainbow zebra was not sick. His genes had just been mutated. The two beautiful zoologists went on to explain the process of transcription and translation. First, in the process of a gene being mutated, is transcription. Transcription is the synthesis of an RNA molecule from a DNA template. So in simple terms, transcription is the combination of an RNA molecule from a DNA mold. Transcription occurs in the nucleus of a cell. Transcription requires an enzyme or a protein that speeds up the rate of a reaction called an RNA polymerase. The RNA polymerase is the promoter or region of DNA that has specific base sequences. It signals the gene to be made. The RNA polymerase binds to the DNA and separates the DNA strands. Using one of the strands of DNA, the RNA polymerase assembles nucleotides into a bonding strand of RNA. This process is called elongation. Elongation has codons, which are 3 nucleotides that sequence the code for a single amino acid. By copying a single DNA sequence into RNA, a single gene can produce thousands of RNA molecules. The process of transcription is ended or terminated by a terminator, which stops transcription. Before mRNA can leave the nucleus, sections called introns are edited out. The pieces left over, called exons, are spliced together. An RNA cap and tail are added to form the final mRNA molecule.
Next, the process of translation occurs in a step of initiation. The promoter signals the beginning of a gene when it goes through the pisi. It then reaches a start codon, AGU, and replication begins. Then, large small ribosomal subunits join to form functional ribosomes, complementary strands attached to codons. Amino acids on the inhibitor tRNA forms a peptide bond with the amino acid beside it. Inhibitor tRNA leaves ribosome behind and a peptide bond forms. Protein synthesis stops when it reaches a stop codon. A mutation may occur when a codon base gets substituted with something else. For example, if adenine gets substituted with guanine, this codes for a certain protein, and if it gets substituted with something else, it may change it. Because of this causes the zebra stripes to be rainbow instead of black and white. This is called substitution mutation.
When the zoologists finished, the parent zebras understood so much about transcription and translation. They now knew that their little zebra was very special. They were so relieved that their little boy was not sick, and for the rest of their lives they lived happily ever after in Africa.